event as a satellite event of the conference, and because this is the opportunity to have in our group, at least for now, the large group of specialists and from many places. To give just a background, we, in 2010, here, we organized a network of modelers in Brazil, of institutions interested in the problem of dengue. So since 2010 till now, we've been meeting every year to share our work on dengue modeling and discussing the future of our projects and what we can do together. So this is an informal meeting. So we're supposed to be in a small room and know everybody talking freely, but we are here in this very formal place, but feel free to, to feel comfortable and say whatever you think. And so I know that some people here will be more comfortable speaking Portuguese and others in English. So those that want to speak in Portuguese, just speak in Portuguese and we translate and we try to share our ideas the best way we can, okay? So we've been developing lots of models and in different areas, from statistical models to mathematical models, and tackling different problems from surveillance to control in the last years, but always devoted to dengue. And the idea of this meeting, of this first moment here, is to discuss how can we use this knowledge to answer some questions in this new problem, that's the arrival of Zika and Chikungunya in the country. And this is the first part of this meeting this morning. Then we'll have a session where posters will be distributed outside so we can see the work of students and things that's been going on. And then at the last part, we'll have a discussion on the current project that we are running. That's the Alerta Dengue. That's the surveillance of dengue that we are doing here in Rio and expanding to other cities. So what I'm going to show here is just a few slides on the current situation of these diseases here. And then Osvaldo there will present some other data, and then we can have a discussion together with Denise and everybody that wants to share the ideas on the challenges that we have ahead and how can we help and collaborate in this process. So everything here is very new to me also. So we've been studying dengue, Zika and Chikungunya, I don't know, for a short time. So we don't have a lot of experience. So I don't have it. So some of these slides were gave to me by Ana Bispo, that's a virologist, and Phil Cruz, that is the head of the reference lab there. So she is the person that's leading all the diagnostic of surveillance, all diagnostic of cases of Zika in Brazil. In Brazil, no, in part of the Brazil. And she was very helpful and nice, sharing some slides, some data with me. So for those that don't know, Chikungunya is a virus from this family, Togaviridae, and it's the same family of Mayaro virus. That's a virus that's present in the Amazon region. And as far as you know, there is only one serotype and 4 genotypes, okay? And dengue and Zika, they are from another family. They are Flavirides, the same way as yellow fever, West Nile, St. Louis, Rocio, many other viruses. And they have We know that dengue has 4 serotypes and many genotypes, and the Zika has 1 serotype and genotypes known. Basically, in Brazil, what we have the 4 serotypes of dengue: dengue 1, 2, 3, and 4. So, in these genotypes. So you have the genotype 5 of dengue 1, the Asian American genotype of dengue 2, the genotype 3 of dengue 3, and and 2 genotypes of dengue 4. And the Zika virus, what we have is the Asian genotype. So this virus arrived in 2014, and there was this hypothesis that it arrived together with the World Cup. But then nobody from the French or from the Polynesia came to the game, so at least not to play. And so there was this hypothesis that they came in this boat, how do you say that, boat, canoe competition, something like that, and that had a lot of people from Polynesia. So that's the thing. So it arrived in 2014, but it started to increase in December, and in this last year it showed as an epidemic. And chikungunya, there are 2 introductions. Okay. One from the north and one into the northeast, with 2 genotypes, the Asian genotype and the African genotype. Okay, so we have the circulation of all these viruses in Brazil, together with the local genotypes, the Janeiro viruses and other viruses that circulate here. Okay. So dengue we know very well. So it arrived the first after a long time without any transmission of dengue. Dengue 1 arrived in the 80s and spread in Brazil, and then dengue 2 arrived in the 90s, and then 10 years later dengue 3, and in 2010 dengue 4 arrived. So we have currently this scenario where with increasing morbidity and increasing mortality due to dengue. And dengue comes in large outbreaks, and they occur throughout the countries. Only the countries in the south, or the states in the south, are really less affected by dengue. All the others have like large outbreaks every year. Okay. Chikungunya is very symptomatic. So it's said that 90% of the cases are symptomatic. It, as far as we know, gives long-term immunity. It's transmitted by Aedes albopictus, and I don't know if by Aedes aegypti, and then yes, too. Through a short incubation period, an acute phase of about 1 week. And the main problem, one of the main problems is that it can evolve to, the disease can evolve to a chronic phase with very severe symptoms, very similar to arthritis, and can be very problematic to people, okay, and to assistants, how to take care of these people for a long time. There are many outbreaks of chikungunya since 2005, and this seems to be attributed to a mutation in the mosquito that made it made Aedes albopictus more competent to transmit it, so it became more easily transmitted. So there are many outbreaks worldwide. And of course, there are many models of chikungunya already. So that's something that has been more studied now. And in Brazil, this is the last map that I had access. So in 2014, there are 3,500 suspected cases, and this increased to 17,000 in 2015. Okay, and they are concentrated in this region, in the northeast. Okay. I mean, they are found in many capitals, the state capitals, the large cities, but they concentrate here in this region, already moving from large cities to small cities. I need to show this because I got very scared of this disease, and I want, at least the Brazilians want to be scared too. This is really something that we don't want to have. And it causes very, in the acute phase, it provokes swelling and a lot of pain, and also severe symptoms in children and all. And in the chronic phase, it can also, these symptoms can remain for a long time. So in Brazil, we have these two introductions, one in the north, the Oiapoque, and the Amapá. Let's come back. One introduction was here in a very remote area of the country, so it's difficult to spread. And the other one was here in Bahia, Feira de Santana, and this is the one that caused a large outbreak with very severe number of, very high number of cases, and like a breakdown of the public health system. Zika, on the other hand, was a mild disease, something that a very neglected disease until last year, and nobody really cared about it. It caused some outbreaks in Polynesia, in this region in the past, but it's not really causing large public health problems. But then, so because of this, we don't know very much about it. Okay, it's as far as we know, it's mostly asymptomatic. But of course, this is based on very scarce information. Basically, what we know is that from previous outbreaks they asked how many people had symptoms and how many people tested positive. And from this difference, they calculate the number of asymptomatic. But of course, there are many problems with this calculation. We really don't know yet the immunity that associated with this virus. So there are some hypotheses that the viremia is so low that may not elicit a strong immunity. That's something that's open yet to debate. There are some problems with alternative routes of transmission. That's something that we can discuss here later. But routes of transmission beyond the vector transmission. And the main problem that brought attention to the Zika infection, the Zika outbreak in Brazil, is this congenital disease associated with the Zika virus. So until this year, we didn't associate any problem, any congenital problem with Zika infection. But here in Brazil, okay, this is the distribution of Zika in autochthonous transmission of Zika in Brazil. So it's already spread in many states. And what I want to say is that the epidemic of Zika was temporarily associated with an epidemic of microcephaly in Brazil. So since what happened is that doctors start seeing babies with microcephaly in women that just had Zika infections. So they start to associate these things, and this led to this association, to this possible causative relationship between Zika infection and microcephaly in babies. And there are many experiments showing that this virus, it migrates to the neurological tissues, and this could cause an infection, could modify the embryogenesis and affect the development of the fetus. So this is the number of microcephaly in Brazil. The number of cases per year, don't remember exactly, but it was like 20 or 30 per year, something in the whole country, and now it increased to 3,000. So it's actually 100 times more than the average. Okay. So this is a lot. Of course, it's a lot of it's a huge public health problems. Okay, that we need to deal with these children that are being born this way. So this is the short summary. We can probably, some of you know more than me, and then we can share some other facts. But I tried to list some of the questions that we could discuss here in relation to these diseases. And I think there are many open questions, some of them related to the natural history of these infections that we really don't know and need for the models, any modeling that we can do. So one question that I share with Denise and Sorgini is why has Zika spread so faster than chikungunya in the countries? So that's something that's not really understood, because chikungunya is more symptomatic, causes more viremia, should be more transmissible, and why is more contained than the Zika. Okay. This could be attributed to many things. For example, the relative importance of other routes of infection. That's something that we can discuss, how to measure and how to ascertain this. It can be associated with vector competence. So maybe our mosquitoes transmit chikungunya not so well, but transmits Zika better. Or it can be an interference between diseases. So we have all these flavivirus being transmitted at the same time, xenophavirus in the same mosquitoes, and then maybe this is interacting. And so it would be interesting to think of models that could help us to disentangle these different mechanisms. So this is one thing that we could discuss. The other point that's important is this relationship between Zika and microcephaly, and what's the expected demographic profile of this association, because if there is an association between Zika and this disease, we would expect a certain temporal and spatial and age structure that maybe if we can derive this from models, we can compare with the data and see if it makes sense, this association that we've been postulating, or if there is something else mediating this association. Okay. And in terms of surveillance, there are many challenges related to how to monitor these multiple diseases in an effective way. So it means we need to think again how to implement these sentinel units, how we collect the biological data, and how many samples we need to have a good representation of the viruses in the population, and how do we use this virological information to inform the epidemiological models or the epidemiology. So this is a combination of virological data with ecological data and epidemiological data. That's something that needs to be done, because it would be very difficult to separate these different time series just from clinical information. And The another thing that it's important is to assess the public health impact of all this. So anytime we devote energy to control dengue, we are taking the effort from influenza and other diseases, and we need to, and this is something that we need to consider. And also, if we are expecting to deal with a lot of chronic diseases and birth defects and all this, we need to know the size of the problem in order to handle it. So there are many questions, from very scientific questions, to try to understand what is this, to very empirical and applied questions, okay, that we could think how to apply our models. Okay, so this is basically this introduction, and I'll invite Osvaldo to show what we can. Yeah, the idea is we can talk a little bit, and then we open to discussion.
Well, sorry, I'm not be able to show anything because the computers are not communicate properly, so we have some problems. Well, I'm very sorry I'm not able to show the data because I brought in interactive form, so I'm supposed to show application to you with the data so you can manipulate the data, but I'm not able to link right now. So maybe later, if anyone like to see that, I have some spatial data on dengue and also some time series on many diseases in Rio, and especially Zika. That's something everybody's interested in right now. So maybe someone start first and we try to solve the problem. I don't know. Yeah, could be.
Hi, I'm Denise Valle. I'm not a mathematics. I'm a biologist. I work with the mosquito. Claudia asked me yesterday night to talk to you. I didn't prepare anything, but I think Claudia also told the major things I'd like to do. To talk to you, just to focus on chikungunya and Zika virus that are the two new viruses that we are challenging here in Brazil. I have some contradictions that I compartilho, that I share with Claudia. Some doubts we have, for instance, we have chikungunya virus. It entered in Brazil in September 2014. Zika virus entered officially 8 months later, on May 2015. We have 80% or 90% of chikungunya cases are symptomatic, they manifest, and only 20% of the Zika virus can manifest. Another thing is that Zika virus has a extrinsic incubation period of 10 days. What does it mean? For the mosquito to be infective, it needs to get virus from some person that is infected. So once it bites this person that is infected, it acquires the virus, and the virus takes some period to develop inside the mosquito and to render the mosquito able to infect someone. This period is 10 days for dengue virus, is 10 days for Zika virus, and only 3 days for chikungunya virus. So the chikungunya virus entered 8 months before Zika virus. The incubation period in the mosquito is 3 times less than Zika virus, and it has much more symptomatic manifestations than Zika virus. So we expected an outbreak of chikungunya this year and not of Zika virus. So this is very strange. And for instance, I talked to the subsecretary of health of the state of Rio de Janeiro 2 weeks ago, and before he told me that before we have all these problems with Zika virus and possible association with microcephaly, all the managers, the public managers, were hoping for an epidemic of Zika virus because it is less grave, less aggressive than dengue. And so they thought if we have an epidemic of Zika virus, we will not have an epidemic of dengue virus, because both are flavivirus, and they also expected that this, we don't know neither. They expect that if a mosquito is infected with Zika virus, it will not be infected with dengue virus. This is one of the questions we have. We don't know. We know that, for instance, the same mosquito can carry both dengue and chikungunya virus at the same time, because one is alpha virus and the other is flavivirus. We don't know if the same mosquito can carry dengue and Zika, but they were beating that the mosquito would not do that. So this is one thing. The other thing is we had recently, 3 December, we had a publication of a group of São Paulo virologists. They sequenced several Zika virus samples, and that are circulating in Brazil, and they realized that these Zika virus, they have some changes in their sequence, not changes in the amino acid sequence, but in the codon sequence, and as if these virus were more adapted to use the machinery, the translation machinery of the humans and not of the mosquito. So all these point to a probable non-vectorial transmission. We also have, for instance, 2 cases in the literature of the presence of Zika virus in the semen of humans. And one, a person in Tahiti that also had very strange manifestations. He had a manifestation of Zika virus, manifestations once, and then 8 weeks later, and then again 2 weeks later, and they found particles of Zika virus in their semen. And the other case that is reported in the literature is two American researchers that were working in Senegal, and they contracted Zika virus, and they came back to the United States, and the wife of one of these researchers got Zika virus, and she never went out from Colorado. So this is, I'm just putting doubts on what Claudia was telling. And so I think the thing more important for us to try to understand is this possible non-vectorial transmission, because in our case, we're telling to population all the time that prevention is important, the vector control is important, but I'm very afraid that we can perhaps be telling people something that is giving them a false security sensation. If I say you have to control your mosquito, but is this the only thing? I don't know. We don't know yet. And the other thing is what's happening inside the mosquito. If one mosquito can transmit more than one virus, dengue and chikungunya, we know, yes, but dengue and Zika, we don't know. Chikungunya and Zika, we don't know yet. So as Claudinha said, up to November last year, nobody cared about Zika virus because it was a very mild, very inoffensive situation. And the other thing I would like to say is that I think in Brazil we have many problems, but I also think we have a very good epidemiological surveillance. For instance, Zika virus entered in Brazil in May 2005, and on 17 November the same year, 2005, we reported to the World Health Organization, we reported the probable association between microcephaly and Zika virus. And the French Polynesia had an outbreak of Zika virus 1 year before, and they only reported 1 week after us, because just they started to look at their registers after Brazil reported this. Of course, we are bigger, but I think we're also paying more attention. We have a stronger epidemiological surveillance. And, well, we have many doubts. And I think it's that concerning the mosquitoes, we would like to see what's going on the non-vectorial transmission. If a mosquito is has 2 viruses at the same time or 3 viruses at the same time, is it possible? How is the competition inside the mosquito? And also, what does each one of these viruses influences can influence the viability of this virus, of the mosquito? Longevity, ability to lay eggs, we don't know. So it's that.
Thank you, Denise. I think we're ready for Osvaldo. And while Osvaldo, walks up to the front, I would like to leave a couple of questions for everybody to think about for the following discussion, inspired but on what Denise said. Is, well, first of all, if only 20% of Zika cases are symptomatic, and we're already so amazed by how fast it's spreading, if we are only seeing the symptomatic case, what is the real size of the epidemic? Where is the head of this wave? So it's one question. And the other question about the interference of the virus within the mosquito. Well, we all hope that they actually fight it out, but I'm worried that we have so many mosquitoes in the field that competition will not likely to be a major restraint. Because, you know, you can have chikungunya on one, Zika on one, and there's still be a lot of mosquitoes left without any virus. So it's something for us to think about. I think we need to look at all possible hypotheses, but we really need to look of all the things that we are not seeing necessarily. So Osvaldo, please.
Well, let's see if we can get now. This idea is to bring you the dimension of the spatial problem. The idea, this is data from the epidemics in 2011, 2012. So let's see, that's Rio. That's the cases in 2011. See, I like to put this map because a lot of parts of Rio are mountains and green areas. So you can see the most part of the city are covered by this 2011, 65,000 cases. And the next year, yes, that's 95,000 cases in 2012. So you can see the whole city, if you can light both together. But anyway, you can see in this 2 years, you have reported cases, I mean, probably many, many times more that actually happen in the city. So, and other idea, important idea is Rio is a big municipality and you have different conditions, environmental conditions. So this area, west area of the city are drier than other part. This area, well, I think we have a pointer here. This area is very dense populated, and here usually nearby Fiocruz where you work, and this area is usually always a hot spot. And so this area is really hot spot always. And here is Brazil Avenue like that. So this is an important road you have, and you can see that all along there's a high density in population. Anyway, you have cases all over the place, and now I like to show you, because the one idea before discussing the Zika cases, we have a lot of other infectious diseases going on in the city. So I just bring one part to just show. Here I have to do some changing the. Well, this is tuberculosis in the city by week. It'd be difficult to do that because let's see if I can. Well, let's try to see Zika. No, well, actually, we have two different informations. You have the confirmed cases and the suspicious cases. This is the suspicious cases. The confirmed cases inside this one. But as you can see, as Denise mentioned, start in May, the first case. You cannot see anything here, but you have a small bump here. This is a weekly cases. But just look at this week. In the end of December, you have 1,079 cases by 29 December. This week only. And the week before, we have 1,200 and something cases in a week. So for sure, you have this delay in notification. So the case is probably going up as well. So this curve is still going on, going up quite fast. And for Rio,
from a disease whose notification is not mandatory, so it's probably worse.
Yes, that's That's true. And we know also that a lot of mild cases or asymptomatic cases as well. So this is something, and that's Rio only Rio municipality. So we are worried about, so it starts in November. Let's say the increase of the case, because here you have 2 in May, 1 here. So it's because other problem is completely mixed with dengue in the beginning. So nobody can really tell apart Zika from dengue at first. So you are everything mixed up. But let's see the
Osvaldo,
Oi?
Considering that the peak of dengue cases occurring in May, April, May, so if this is the same pattern, we'll have this increasing until May, probably.
Yes,
Yeah,
well,
okay.
could be. I don't know. I think everybody's really concerned, but it's really going really fast. And I don't have, I didn't analyze the spatial pattern right now, but we have the data. But the point is we have to georeference every code and take a while to have this data geocoded. So I think we have to wait a little more to at least for 2015 and then starting to the next year. So I think it's everybody's really worried about that. Sorry? Oh, I don't have the dengue here because of different systems. That's the Sinan, and dengue has a different system. There's a national system. So I don't have here because this here is supposed to be only Sinan. But as I mentioned, I have other diseases here. It's interesting to see that other things are going on in the city at the same time. If I can, let's see. Yes. Yeah. No, that's not a good one. Spikes, but leptospirosis I was looking for. Eu não consigo ver. So see, you have leptospirosis going on, and the symptoms can be quite similar to dengue in the beginning. So it's a potential confusion. Seems to be going down, but we never know. It's because everybody's reporting as a dengue in the beginning, and then only the cases get worse. Then you realize that's dengue, and so on. So it's well, there's many different. There's and this is a thematic disease as well. In the beginning, people are reporting Zika as symptomatic diseases. So you have a kind of spikes over there as well. Not supposed to be, but so only May they really starting to get into Zika data. So questions?
Okay, so maybe we could open for discussion and questions, and maybe Leo, you want to say something? Okay. So let's be informal and just share some ideas. Or,
those notifications they're not mandatory, right? So the Zika notifications are not mandatory or compulsory, but so I'm just worried if that drop down, because I don't think it's delay, because the week before you have the same delay, so it's not a delay problem.
2 to 3 weeks to get the data, and that point was 29 December. And consider that end of the year, you also have holidays and things. So the notification could be.
One thing I was just thinking that people just don't bother notifying this thing, because it's like for non-pregnant women, it's like a mild disease, so it's just a waste of time. for something that's not compulsory? I'm just wondering.
Yeah, but I feel that the population are kind of worried about. So if people had mild or a little bit like the head spots, a little bit fever, they go to the doctor because they see in the press, they see on TV that's the new virus and things like that. So in part, I think that could happen as well, but maybe not now. I think now people are kind of involved and ask about, and the population are usually so, but yeah.
Let's move on.
Well, one thing that maybe we could use our models, our knowledge from dengue dynamics, is to see how, I mean, we can think if the dengue and Zika are transmitted in the same way. Assuming this, we expect the same seasonal pattern between the two diseases. So maybe we could use our models to, or dengue models, to see what would be the expected temporal distribution of Zika if only the vectorial route was being used, and then compare with the data. That's something that maybe we could do. Yeah.
You say that in the case of Zika, they suspect about other routes of transmission, and you talk about SAM and things like that. Denise, they test, for example, the husband of these wives that were having babies with problems. They tested them to say that the woman was positive, the mother. They make something related to the father of the baby to say if it is positive also.
I'm not sure. I think no. This is some suspicion that is being raised now. And the other thing is that I'm not quite sure that we have a strong and specific diagnostic tool for identify Zika virus specifically. Everything is very, very new, really.
A problem related to that is that the only really specific way we have to identify Zika so far is by PCR. And Zika, I mean, it's strange in many ways. One of them is that apparently you make, in the beginning of the infection, you make a small peak of viremia when you compare with dengue, for example. And then the virus, for some reason, nobody knows, it disappears very fast from the blood. But it's still there because you can find virus in saliva, you can find virus in other secretions, but it's hard to find it in blood. So ideally, we should search for antibodies against Zika to determine if the person had dengue, had Zika or not. But we don't have a serological test available yet. I mean, we have many labs in the country working on the development of such a test, but we don't have nothing validated so far. So it's difficult to do what you're saying because probably you won't detect the virus from using PCR when the other person starts showing the symptoms, and you don't have any other tool to do that. But I think that one important thing that points to a non-vectorial transmission is the fact that when we go to the bench and we try, for example, to infect the mosquitoes with chikungunya, basically we get 100% of infection. All mosquitoes get infected, always. And when you try to infect mosquitoes with Zika, I mean, we don't have many groups that have done this so far because, I mean, a few people have the virus to work, and that's a problem. But it's very hard, apparently, to infect Aedes aegypti mosquitoes with Zika. You need to use very high virus titers, and we know that this is not what happens in nature because apparently don't make high viremia with Zika virus. So Aedes so far, and all this is very preliminary, but so far Aedes seems to be a bad vector for Zika and a very good vector for chikungunya. And still we are seeing Zika spreading much faster than chikungunya. So doesn't make any sense. It really strongly points to a non-vectorial way of transmission. And I think that eventually, well, I'm a biologist. I don't know mathematics and stuff like this. But I think using the knowledge, besides what Claudia already said, I think that using the knowledge that we have on modeling dengue spread, we should be able to show that the speed of transmission, the high rates of transmission and the high number of cases is not compatible with a transmission by a mosquito.
May I just add something? I would like to talk a little bit about a vaccine, the Sanofi Pasteur vaccine that has been liberated now. I don't know if you listen to this in the newspapers and so on. It protects 66% only. And my concern is that it has not been tested in the context of an environment where Zika virus is circulating. And Zika virus is very similar to dengue virus. Some virologists say that it could be also the 5th dengue serotype. So what will happen with persons that have been vaccinated for dengue virus that protects you against the dengue 1, 2, 3, and 4, and not Zika? Will I can see 3 possible scenarios. Nothing will happen, it will protect, or it can exacerbate the Zika manifestations. I don't know. So, is that?
I have a question for entomologists in Brazil. I work on Chikungunya in La Réunion, and in La Réunion there is only Aedes albopictus, and it turns out that albopictus in La Réunion, at least, has a very good vector for transmitting chikungunya. And so, what is this? I don't know the situation in Brazil, but do you have only Aedes aegypti or albopictus, or in between, or something?
No. Basically, we have both mosquitoes in Brazil, both Aegypti and albopictus. But for example, for the case of dengue, at least, albopictus has never been incriminated as a vector. We know that in the lab it infects, and so far the same is true for chikungunya. We know that in the lab you can, chikungunya is an alpha virus. And alpha viruses, they usually infect mosquitoes very well. The same is true for Mayaro, the same is true for Sindbis. And all the alpha viruses we have tested so far, they infect very well. So, and chikungunya is not an exception. So it can infect Aedes albopictus. We have done this in the lab, but so far, no one has incriminated it as a vector, only Aegypti. Mm-hmm.
From La Réunion.
I think it's also because of the strain of chikungunya that is circulating in Reunion Island. It suffered a mutation, and it is more efficiently transmitted by albopictus. And here in Brazil, we have both strains of chikungunya.
So, is there any other people in the audience with experience with chikungunya modeling and all? No, but please, I was wondering if we could share some ideas.
Sorry, no, I raised my hand before you said the chikungunya thing. So I wanted to know, because dengue is so complex immunologically, does anybody know how Zika sort of manifestations might be affected by previous exposures to one or more dengue strains? Has anyone looked at that?
Actually, this is one of the hypotheses for this increasing microcephaly, all these developments. That would be an interaction, somehow, some sort of interaction between a Zika infection in a host with a dengue immunity background, or Could be too. So we really don't know. There are some case control studies starting to be developed and cohort studies to try to understand better this relationship between dengue infection and Zika. That but really is yes, is an important question here.
Just two comments. I really like the idea of using the models with whatever data you have available. I realize it's not too much, but you have the spatial spread and you have some of these initial epidemics. The two unknowns are of course, the asymptomatics and then this speed of propagation, essentially whether you have a vector or not. And if you assume you do, of course, it's a matter of how quickly that transmission through the vector. So whether you effectively can differentiate that by fitting models to some of the data, it's an open question, but it may be worth seeing if with some of the assumptions of what you know about the mosquito, you would get very, very, what kinds of parameters you would get. So I think that could be an interesting exercise and see what trade-offs occur with the perhaps the rate of asymptomatics or underreporting, even if I realize at this point the data on Zika is not very much, but I think it would be an interesting exercise to try, at least on those two unknowns, right? But on the other hand, I see, well, I was curious because there is a lot of discussion here this morning on Zika, and it seems almost as if you say we have models for dengue, we more or less understand dengue, but it seems like there are a lot of, the dengue is still the major, you have Zika as an emergent problem, but dengue and the way it's growing is an enormous problem that is by no means solved in terms of understanding its spatial temporal dynamics. So I was a little bit more as a question because we saw a map of what happens in Rio, but that's accumulated cases. And of course, there must be some temporal dynamics to that is of value to public health. And I know some of you work on that. So I was more interested in here well, I don't know anymore which verb to use. Yes, what do you think are the open questions in your work on dengue and the unknowns for Brazil in dengue? Because it seems like it's going up in prevalence and it's going to be an immense problem. Of course, there is the wonderful Wolbachia prospect, but that we saw wouldn't spread very fast and would require also a long effort. So I'm curious, I mean, I know you have good models and one interesting question that comes up in terms of the modeling and the four serotypes is the recent publication that perhaps the 4 serotypes are meaningful genetically, but are not meaningful in terms of distances in cross-immunity space. There was essentially an antigenic map published this year showing that perhaps the consideration of 4 discrete serotypes for modeling cross-immunity may not be meaningful. So anyhow, that's of course perhaps at the moment a more theoretical question, but I was interested also in the spatial temporal dynamics within cities.
From my perspective, I think that something that's specific to our scenarios here is that we are still seeing the transient phase of introduction of dengue in the country. So if you consider that the last dengue virus arrived just 5 years ago, so it's just caused an epidemic in 2012, like 3 years ago, 4 years ago. So you can think that this is still in the process of reaching a steady state. So one question is what this steady state will be. So and also you've been very bad at predicting what the next serotype will be. So For example, we know that, okay, this year dengue 1 is emerging, but what's the consequence of dengue 1 emerging after dengue 4, dengue 3, and dengue 2? So this is something that we still don't know. And also how to integrate this virological and genetic data really with the epidemiological data in order to really better measure the spread of the disease in the country and make predictions. This hypothesis that of course, we know that dengue is very seasonal, but seasonality changes from one place to the other, and because the climate varies in the country. But we tend to see that when the dengue season starts earlier in the year, it will grow larger, just because you have a larger season than. So we have this many facts, but it's not really within the models. I would say that there are many things that we see, but it's not really modeled well yet. So we think I would say that these are the main issues, but maybe Oswald wants to say something about the spatial pattern.
Well, I think the one of the problems, we don't have baseline on serological state of the population. I think that's a kind of big problem, because dengue 4 was kind of easy to predict somehow, because you know that nobody have immunity for that, so it's spread like. But right now, I'm concerned that in parts of the city you can have different virus going on, but you don't have enough sampling on virus to really assess that. And I think that's one of the major problems we have right now, is to get more data on a kind of serological surveillance, what's going on, not only dengue, Zika, but other viruses that go on in the city at the same time. You have the influenza, and you have many different things going on, and we need to know what's going on. And actually, we don't know right now. We only observe the surveillance systems that only capture a fraction of what's the really dimension of that in the city. So, and the spatial problem as well. I think that's a city that has a strong connection. People travel a lot to go to work and back and forth, not only in Rio, but in all the metropolitan area. So it's not only a municipality or a city, but it's a complex of cities interligate together as a metropolitan area, and that's a lot of commuting between all the parts of the city. So I think that makes sense to think not only in Rio municipality, but the way the health systems in Brazil are located, the surveillance system is each municipality take care of their own systems, and the quality information is quite different between them. So we have a lot of problems to get the data from the other municipalities, and the quality of the data varies a lot. So, and other point is that it's very difficult. something like 23% of the Rio population lives in favela areas, and you don't have formal addresses. So it's really difficult to pinpoint the geolocation of that. And sometimes you can identify that they live in the favela, but sometimes even that, because the name of that street doesn't exist, because in the community they used to have a name for that street, but that doesn't exist in the official databases. So it's really hard. So you miss a lot of geo-reference the cases from these areas. So we have the cases, so in the temporal aspect you can retrieve that, but not in the spatial. Even so, we have a lot of science, let's say, we have a lot of data, and we could try to do some analysis and things like that. But I think it's a big point is really introduce this commuting data and traffic data in the city to allow, because one of the problems is that we are only using residence address. So, and the people, get all over the city, and we have some data right now showing the displacements, and so maybe use that as well to understand more or less how it works.
Currently, I think that in the last months, what we've been doing in practice is thinking on how to collect data. So, so this involves the design of studies, so epidemiological studies, and but I think that we can use the standard epidemiological methods, like have cohort studies and case control studies and all that, but it would be interesting to see what the models need, the specific models need, because sometimes we can actually try to change somehow these standard designs to introduce these other variables. So that's something that will be very important at this moment. Think of what we want to measure in order to feed the models. And in some situations we have, now we have many projects starting, epidemiological studies, entomological studies and all that. And this will be an opportunity to try to fit in this our models somehow, and but for this to happen in a successful way, we need to come up with ideas on how to measure the things that we need to measure. So that's something that we could take from this meeting, try to think of this and see how we can collaborate to improve the experimental and the epidemiological studies in order to fit the models better. Okay, so. Okay, so one thing that I would like to have as a result of this meeting, of course, we are just talking a little bit about the subject, but if we could organize groups that have common interests in these topics, we are very open to collaborations with everybody. This is an open network of modelers and researchers interested in this, in Zika and Chikungunya and dengue. And this is an opportunity to test models and to explore these relationships, and also try to help to deal with the situation that's really scary for us that are here. Okay.
Don't have enough, perhaps, information on serological information, and then you say, well, how can we plan? What do we need? Because it seems like in dengue, we saw yesterday, even in Bernard's talk in Thailand, where there's tons of data, they continue. I mean, there is a continuous debate of, whether the size of epidemics is mostly determined by some of the serology or the climate variability. So the models haven't resolve that. The statistical analysis haven't truly resolved that. So there was, for example, a recent competition set up by NOAA on prediction. Maybe some of you participated with the data from Puerto Rico and something, and it showed some of the limitations. And so it's still incredible with all the model that this interaction of climate and serology in that disease continues to be, I think, a fairly open area. So the question of sampling, at the moment, the question of sampling the diversity of the virus, it should be easier and easier as time passes, and it's an area that I wonder whether you can use the models to kind of, or your perspective as modelers to somehow convince the public health system that this is a priority, and what would it take to do that? Because it seems like we we can't tell at the moment, should it be enhancement, should it be cross immunity, how should you model that? So you can ask the question about Zika, but you don't even know about dengue. So I think it's still remarkable, and using perhaps spatial information to get at that question at more the micro scale may be really interesting. So I was wondering what would it take, because Brazil has, compared to many other places, pretty good surveillance at the moment, and but I wonder, and also lots of work in virology or virology. So how to couple those efforts perhaps more effectively? Whether I think that's a really interesting direction.
I would like to, in response to that, I'd just like to share a little bit of our experience in the last few months, because we're going to talk about this later on in this morning, but we have this joint project with the health secretaries on our Rio de Janeiro state, and we managed to develop a really nice relationship with them. And one thing that was key to this nice relationship was that we took the responsibility and the responsibility to provide them with analytical results on a regular basis, so that they can do their job, better. And I think that we could take this relationship further. We're still trying to, unfortunately, as much as we have a very good surveillance system, the health management is very segmented. The states are pretty much have their own controls, each state, and then there's the federal government, which integrates data but does not really control things at the end. So it's going to be an uphill battle for us to reach a larger portion of the country, but we believe strongly on this approach, which mainly is a partnership between scientists and people who are in the field trying to combat the disease, because they want partners, they want help, and we want data. So they are willing to give us data as long as we are willing to give something back. So I think it's something that we need to keep in mind, and it has worked for us, and I think it can be expanded.
Well, she mentioned about the serological research. So we are involved in a project that we want to estimate the distribution of the serotypes and Zika and Chikungunya, all the dengue-like disease that are happening in Rio. And we design a sort of a simple experiment to do it, and we are a sampling for surveillance that is going to happen here in Rio, and spatial surveillance as well. But if you look at the history of the data, the serotypes for dengue is very poor, very poor. I don't know. Last year they did 70, I think. 70 during the epidemics. They have some 70 in the whole year. Yes. So just 70. So well, that's nothing. So basically, we are trying to do it, but it's expensive. So we designed the experiment, but we hope that they are implemented. I don't know, because it's expensive. So I don't know. It's 500 samples per month, and taking a PCR for each one. And I don't know, not one PCR. We have to test for Zika and dengue and all sort of things. It's going to be expensive. And well, I don't know if they buy it. They say they will do it, but you never know. So yeah. So well.
Okay, so we can stop here, go to see the posters and coffee break. I just want to say that we can take the discussion to the more small groups and think about it. And but the idea is that we really can try to collaborate in these things and try to think together in this. And these are great ideas, and I thank you for the suggestions. And so we go to the poster session, then we come here back in a while.
Não, minha irmã. Eu.
Como é que fica sabendo dos vídeos institucionais?
De nada. Hello, everybody. Welcome back to a second part of our dengue meeting. In this second part, I will invite some more colleagues. We will focus on various aspects of our ongoing continuous vigilance monitoring surveillance project that we've been conducting for over a year together with the Secretary of Health of the state of Rio de Janeiro. So I will invite many people, actually 3, to talk about this project. Okay, so let's get started because we're already very late. So I will invite Cecilia first to. Is it that the order that we planned? Yeah, right. Oh, Thais first. Okay, sorry. Okay, sorry about that. Thais will go first, then Cecilia, then Marcelo. Okay, Thais, you have the microphone.
Well, good morning to all. So I will try to speak in English and probably sometimes I will speak in Portuguese. So I will talk a little bit about this. The beginning was a project and the aim was to construct a website, a product where all the people interested to know what is going on in the city where they are living about dengue, the situation about dengue. And this is a beautiful work. And I think that we could reach our aim. And now we are working to be doing this bigger, and then we could reach all the country. Is a dream, but we have to dream. So yes, no, next. Ou tem como? Esse aqui? Como que? Esse aqui? Sim.
So what is the InfoDengue alert system? It's a system with generated update alerts about dengue cases in a city. And the most important is that this system is available online and it's open. So anyone who wants to know the information about dengue can reach this information. And I think that this is the most important because sometimes people in a city, they just knows information that comes from television and news, but they really don't know what is happening in the city in real time. And with this system, they can reach a map of the city where we are trying to present the situation about dengue in weekly. And this is, I think that this is very important because this brings some kind of power to people in their neighborhood because they can see what is going on in their neighborhood in that exactly time. And maybe they can try to looking for ways to change the situation in that area. And the system works with a set of mathematical and statistical models. And these models, they are fed with data that came from Twitter. And this is new in this kind of surveillance program, maybe I could say, because we are trying to understand what people use the data that is generated with people that talk about dengue every time. Because most of programs use only the official data, but people is in real time is talking about this all the time. They are talking about this, and they are talking about this just because it's new. It's a new, or is because something is happening in that place where they are living. And this information, it's very important also. So we use epidemiological data, that is this notification data, official notification data, meteorological data, and in some cities, so these three first, we use it, we are really using use it in the system. But the entomological data, we are working to insert this information. And now the cities that works with, that generate entomological data, we can analyze this and separate it and bring this information to the government, the city that is using, and they can use this information to their political choices in the city. So, como é que usa isto? Sim. The InfoDengue alert system works in this way. So, so we use this. The tweets has suffer a kind of a filter to choose only the words that really have some kind of that meaning something to this dengue situation in the city. And we use the officially notified dengue cases. And these notified dengue cases, they have a kind of a delay between the day where the report is done and when we receive it, the information. And there is a delay between the cases. They spend some time to type this information and to bring it and to become available, this information. So we have to do a correction in this delay. And we use the meteorological data. And the meteorological data, it's very important because this data have impact in all the vector, the population of vectors in the city. So we have this the rain and the temperature and the humidity. They have influence in the vector density, and the vector density has influence in the off position, and in this fed each other. This because if you have too much eggs and you have good conditions, meteorological conditions, this can bring a new population of mosquitoes in a place. And the vector density is related with the dengue incidence. And the dengue incidence is the officially, when we have the cases of dengue, we have the officially notified cases, and we have people talking about dengue in a place. So with these three components, we can generate the alert system, and with this alert system, we can generate weekly reports, time series about the dengue situation in a place, and we can generate maps where you can see the situation of dengue in different areas in a city. So this data we use, the Twitter data is available from a partnership made with a group from Universidade Federal de Minas Gerais. The name is Observatório da Dengue. This is the website. And they have this project also, this program, where they bring this daily, the update is real time also. And they can show in this website what people is talking weekly. So it's weekly. And they shows what people is when people is talking about dengue in weekly, they are showing this. And then the meteorological data we use, basically the data we can take in this website named Weather Underground. And in this website, we take basically the data, the official data that came from airport stations. And the data about dengue, this is maybe this is the most important part of the system, because the dengue data we use, it comes from a partnership between us and between the municipality or state health secretariat, because the data about dengue that is available from federal systems, there is a big delay between last time I saw in DataSUS, the last year you can find data is 2012. So we need data from now to know what is happening now. So it's very important, this partnership we do with, we did with these secretariats, because they sent to us weekly the data, the up-to-date of dengue. Every week they are sending this to us, and we use this data to generate the maps and do the feedback to them, explaining or trying to explain what is going on in that neighborhood, in that city. So this data has a huge number of columns, and but we only use this group of, more specifically, this group of data in this database. There is the data of notification, epidemiological week of notification, year of notification, first symptoms, the register, the epidemiological week, and the name of the neighborhood. It's important to say that we don't use nothing that can connect the cases with the people that live in any neighborhood. So there's non kind of identification of the people. This is it's very important. And then this is the like the website looks like. So if you, and now we are going to open the website. And well, oops. And go. So if you see, this is the last updated was Monday, yeah. And if we can see all the city, you just só pouquinho para cima, obrigada. Desce pouquinho. If we can see all the city, it's yellow, is in a kind of attention warning situation. And this is this division of the city. It's a division where the Secretariat of Health do the division of the city according to groups, the health groups, like poderia dizer assim, and if we see like this, all the city is in notation. So each point of this, você pode colocar só o mouse em cima de pontinho verde qualquer desses. Isso, qualquer para abrir, né? Qualquer pontinho você pode clicar. Isso. Well, each point of this is the localization of one health center. So the people that lives in this area and see that, oh, my area is not that good about dengue and I'm not feeling good. And if they look the information here, this person can look for the most close to their house, the most center of health center more close to their house. Here there's the address and the name of the center. And if we look, we click in this. Here. Isso. Well, você apaga as duas caixinhas, depois abre uma. Isso. So if we see, you can click, and then you can choose if you can just only, if you only can find looking for the health centers or the areas of this division of the city, all the areas. And then here in this part, there is an we can explain which means each one of the alerts of the colors alerts. And here we try to show the estimated cases in the present week. We did the alert. And os níveis. So in this alerts, the green one, when all the map is green, means that we do not have risk of dengue in the city. And when we have, and this is the first and the second one are more related with temperature and more climatic factors. And these two are more related with the dengue cases. When we start to have dengue cases and we have this situations about, or when we have more than 1% of account, we do more in this area. So we have an orange situation. And when we have more than, could be more than 300, Claudia? When we have here, it's more 100 to 1,000. And then in the yellow is when we have a dengue season, and we need we have in this situation we have climatic conditions to reproduction of the mosquitoes, and there is a great possibility to start to have transmission of the disease. And in orange we already have the transmission, but
Yeah,
it's
interesting to point out that at the orange level, the way that we transition from yellow to orange is that we have we have a continuous estimate from week to week of the effective reproduction number. So when the effective reproduction number of the transmission goes above 1, we calculate it, confidence intervals as well. But when it's significantly above, then we switch to orange, and the red level is it requires for you to at least have sustained transmission. But then when you go above a threshold of incidence.
Okay. So wait. And oops. Você pode voltar para a apresentação? Mostra as séries também? Ah, tá. Então espera só um pouquinho. Ai. Tá no site ainda, aqui em séries históricas. So here all people has the access to the historical series about temperature, tweets, and then cases in each one of that areas of the city. It's available, this information, and here's a series from 2010 to now. And here in this topic, if you click here, you have the possibility to do the download of this series if someone needs this information. And this is, well, it's available for all series, all the regions of the city. So, pode voltar para a apresentação, por favor. So this was the series, and then to the city here in Rio de Janeiro. This project started with a partnership between us and the Secretariat of Health of the Rio de Janeiro, and it's working. It's the website that is working now, and weekly we send them this report telling about information. There is information about here tweets and case of dengue and temperature. We send data about the last 6 weeks, what's going on since 6 weeks ago to now. And there is also this map. And to all of these areas, we presented some series telling what it's going on in all the time, during the time. And now we did it, we are working, under implementation. We did a partnership with the Secretariat of Health of the state of Rio de Janeiro. And today we could, by now we were able to include 28 cities of the state of Rio. This is the map. This is still not available online, but we are working for this. So in the same way, we send weekly this report to the Secretariat, telling about and a small overview about the situation in that week. And here we separated by regions of the state, and the person which is responsible can choose the city, and they have the information about the time series from 2011, 10, I don't know exactly by now. And they have to also, we also included this table with the last 6 weeks information about the cases of dengue and all of this. And so we are, so now, like I said, we are working to include new cities from Rio de Janeiro state. And the problem is that most of these regions, they are not, the data from airport station don't reach these areas. So we are having some problems to get a good meteorological data, and now we are working to get some satellite information about climate in these regions. And we are under negotiation with the cities of Campo dos Goitacazes to generate an alert to this city in special. The city of Paranaguá in Paraná State today is suffering with epidemic of dengue. So they are very interested to make part of the system. And we have been talking with Porto Alegre. There is an interesting city because they have few cases, and it's very easy to see when the cases, when there is the beginning and the end of the cases in the city. So we can follow the cases of dengue in these areas. And coming soon, and we are working a lot for this, we are leaving available in the website with a multi-city website where anyone which is interested could be open the map of the city that this person, the choice of this person, with the series and all the information weekly about dengue of the city. So it's this.
Thank you, Thais. We are now going to go forward with Cecilia, and then after Marcelo, and then we can open for discussion and hear your suggestions. And this project is it's a good point to point out that we are always looking for partners. If you're interested in help with analysis, if you have ideas of how better to use this data to provide other sorts of benefits for the population or for the health control system, you're very welcome to talk to us and become a partner of our project. We are not at all closed. We are always looking to expand our research group.
Well, thank you. My name is Cecilia. I'm going to present to you a little bit about my work in progress with tweets. Actually, in my PhD, I'm working with Porto Alegre model of dengue introduction. But at first, we decided to work a little bit with tweets because it's important data that we have, but we actually don't know what is the importance of tweets. So we are trying to present to you a little bit about this. How important is tweets for this kind of modeling? So at first, I am trying to understand a little bit better how can tweets be used as a risk indicator factor, and also if we can use tweets as a good proxy to our models, considering that dengue cases is a difficult it's a difficult variable, it's a difficult information to get. So could we use tweets instead? So this is the kind of question we are trying to answer. Just to show you a little bit how these algorithms for tweets are done. So at first, we have two kind of processes. The tweet data passes through two different processes, and this one is to collect information from the huge database that we have, and the other one is to differentiate the information that we have. Since you can imagine that tweets have all kind of information inside, Like we have personal experience information, which is the one that we are interested in. And we have like propaganda and marketing. We have information like campaigns from the municipalities and all this. We have opinions, and we have like jokes and irony. So we have all kind of information in tweets, and we may not use all of them. So we are interested in personal experience information. And just to show you a little bit, when we consider this personal experience information, we know that they are the most important information correlated to dengue cases. But also here, I'm not sure if you can see, this personal experience one is the green line here, and this purple line is the complete data, is all data. And even though we know that the personal experience data are the most important, we can see that it's very, very close to the correlation of dengue that we have with all the information. So we will use the personal experience, but it's very close to the complete data that we have. Just to show you a little bit about tweets in Brazil, what kind of data that we have. We can see here in B we have, in A we have the tweets. Here we have dengue cases, and here we have population. So you can see that we have a good correlation, or the tweets are in the cities that we have the biggest populations, and somehow correlated with the cities with biggest dengue burden or biggest dengue cases. And we may guess that tweets are very, very correlated with population. So biggest cities tend to have more tweets. We can imagine that. But also the city with a lot of dengue, we have some correlation, some good correlation with tweets. So we are trying to understand better this kind of correlation. But just to show you that we have a strong signal of tweets here in these cities that we have the more importance of dengue. Here I did some comparison between tweets and between Google Trends. I think you may heard about Google Trends. The difference is that in tweets we are considering information from some individual. So someone have to say something about tweets, about dengue, sorry. And when we consider Google Trends, we don't have an individual, we have news. So it's all kind of information from dengue or Zika or whatever. Here I am comparing dengue tweets with dengue information with Google Trends with dengue information. But just for you to know that one of them is about individuals and the other one is about news. So at first here, I have a graph with the gray bars is the tweets, and the red lines are the dengue cases, just to show you a little bit how they correlate. Here I have from 2012, August 2012, when we start collecting data from tweets. And here we have May 2015, so almost 3 years of data. And we can see here that tweets, I didn't show you before, but we have tweets, signal from tweets from around 200 cities in Brazil, 200 cities. So this information from tweets in the bar are from those 200 cities, and this red line here, the smaller one, is the dengue cases in the same 200 cities. And in the other line, the bigger one, we have total dengue on Brazil. So we can see that they correlate well. They seem to have the same pattern, but as we can imagine, considering all the dengue cases in Brazil, we should have more dengue cases. But here, the comparison between tweets. So I have the blue line here, the tweets, and the green line here, the Google Trends, and the gray bars are dengue cases. So at first we can see that they the both of them have good correlations with dengue cases, but I don't think you can see here. Google Trends signal is very, very small, but still we can see that it increases when dengue cases are increasing. So both of them can correlate well in this increases. And here we have absolute data from Google Trends and from tweets. And here we did a first analysis of prediction, considering only the data from tweets and only the data for Google. No dengue cases, nothing like this. Only prediction of dengue cases with a linear model, very simple, just to see what kind of information that we had. And you can see here that, as we could imagine, tweets have a bigger signal, a little bit closer to dengue cases, and Google Trends we have a smaller signal, but both of them following almost the same pattern as dengue cases. Just to show you that they both correlate well, but we have to consider that Google Trends only have information for the whole country, for Brazil, and it is not a good amount of data. But with tweets, we have a good amount of data for Brazil, and we have data for the states and for the city. So to have a better comprehension of tweets and correlation between tweets and dengue in the city level, we have to use tweets, at least for now. So starting to work a little bit with those 200 cities that we have, data from tweets. I'm trying to divide the cities in different groups to understand a little bit better how are these cities related. The cities that have the best correlations between tweets and dengue, what are the characteristics of the cities? So at first, we are trying to divide the cities in these 4 different groups. I will show you a little bit about the group that we are considering as high quality of correlation between dengue and tweets. And here, at first, we are considering cities with the correlation, a simple person correlation, bigger than 70%, and no zeros. Here, I think it's easier to explain here. These are my timeline, almost 150 weeks. And I'm considering that I have high quality of data when I have dengue and tweets in more than 100 weeks. So we have almost, I have data from almost all my time series. And here I have the correlation, just to show you that in the biggest part of almost all my cities have correlations that we can consider as high. But I will show you a little bit just about the group that I'm considering high, which is these 3 groups. This is the distribution of the cities. As I said, the groups that I'm considering high, it's distributed around the country, but since I'm considering cities with high correlation and a lot of weeks with data, we could imagine that the Southeast would be the most represented region. So most of the cities are in this area, but we have some city represented in the other regions of Brazil. I think you cannot see what's in here, but you may believe me. These are the 39 cities that I'm considering for now as high quality. The idea is to publish all this data for anyone who wants to study this data and to check if the city is considered high or no. Especially to say that here we are doing general analysis of the city. So if you like to improve the modeling for each city, it will be great for us to understand better how the different cities could be improved in dengue modeling. But just to show you a little bit that we have like 10 different states of Brazil, considering this list. So it's very well distributed. We have populations from like 100,000 people to 6 million people. So it's very, very well distributed. And we have very good correlations considering just a % test. And we have, and here we are trying to understand a little bit better from the cities, how can we better model the cities? Again, showing a little bit about what are the characteristics that we have that differentiate these groups, considering for now this differentiation that I have for now, but we can see that there's no statistic difference here, but these cities that we are considering good or high correlations tend to have biggest populations, have more access to personal computers in the houses, more access to internet, as we could imagine, and they have the biggest signal for tweets and dengue. So this is what we could imagine. Just to show you, I chose I chose 3 different cities from the group, from my high quality group. But just to show you how it's working. For now, we are using simple modeling with linear regression, but we are trying to improve this using autoregression and some other tools to have better modeling for the cities. But even though we have the simple model, we can see that we have great correlations between the dengue cases and the tweet signal. We have strong correlation, strong linear correlation, as I showed you before. And if I consider this only tweets as a prediction model, we have a good fit between the dengue cases, observed dengue cases, and the predicted one. Just to show you a little bit what we are trying to show. We have this information for all the cities, but just to show you a little bit of what we are studying, and we are trying just to show and to integrate a lot of different studies and to prove that tweets is a very easy to get information, and it's good to a lot of different cities. So the idea is to try to connect more people to work with tweets, and especially to have have different studies developed in the cities to have even better models. And one of the ideas is maybe we could use tweets as a good proxy for dengue cases, since we have a lot of trouble to get this information and the delay in notifications. So maybe we could use this kind of information in the models, which would make a little bit more easy to develop real-time modeling for dengue cases. Well, that's it. I'd like to thank. I would like to thank some of our partnerships, and thank you.
Thank you, Cecilia, for an excellent presentation. Now I'll call Marcelo Gomes, which is going to talk about the future challenges for the alert system. That's a lot cooking in the oven, so he's going to tell us a little bit about it. Marcelo.
Hello, everyone. As Flavio has just mentioned, my talk will be about future next steps and future challenges facing the Alerta Dengue project that we just heard about. And so to start, I'm going to list a few, some of the things that we are aiming at and what are some of the challenges that we expect to face and how we want to face them. Okay, so first, let me talk a little bit about OV traps that, as was mentioned by Thais, is one of the one type of data that we have already for some cities, and we are currently not using in the alert system yet, but we intend to. And now one of the challenges with ovitraps is somewhat similar to what we face with temperature when you use temperature information, is that we have to look out for that. To have ovitraps or data related to ovitraps is not necessarily the same thing as having good data for what we want to do. Okay, because there's difference between quantity, and quality is purpose dependent. Okay, it's one thing to have ovitraps that are good to estimate mosquito population in a given region, and to have this data that is useful for the mosquito population in transmission areas. Okay, I'm gonna go through a little bit of detail in that using Rio de Janeiro as an example. Okay, so just before continuing, this is not what is the current situation in Rio. I'm just going to use it to illustrate what I mean about the difference between quality and being purpose dependent. So let's suppose that in Rio de Janeiro city, so this is a map of Rio de Janeiro city, you have here some of the neighborhoods, and let's suppose that most of the ovitraps in the city are in this area here. This is the Tijuca National Park. Okay, which is a very dense green area. And so imagine that most of our ovitraps are placed here. Now this information would be very good to estimate mosquito population here, maybe here as well, but it's not necessarily good to estimate mosquito population in those areas. Why? Because here in those areas we have a completely different environmental conditions. Okay, so it is to be expected that how mosquitoes are doing here is not necessarily the same thing as how mosquitoes are doing here, because the environmental conditions can be completely different. Okay, and to our specific purpose, which is to study transmission of dengue, in those areas is where the transmission is most likely to be occurring. Okay, it's in the urbanized areas and not on the parks. Okay, so this is what I mean by having the challenge of knowing where those ovitraps are to then estimate how good they are or how useful they are for our project in particular. Okay, and we have already, this is an ongoing discussion. There are people working on developing models to better establish what would be a good ovitrap distribution in a city that would be useful for our for this particular idea of using ovitraps to inform dengue situation, okay, and dengue transmission or vector transmission in general by mosquitoes. Okay, so this is one of the challenges related to ovitraps that we are likely to face. So next, another important thing that we want to do is, since Thais as well has already mentioned, we want to expand this project to different cities and we incorporate more cities, more areas, more states. One of the things that we want to use here is human mobility as also another variable in the alert. Because if, for instance, if I have a situation where I know that I have a lot of cases already in a given city, and on another one that is currently there's not much going on, but I have a huge flow of individuals moving from one place to another, it is to be expected that this other place can be facing an outbreak soon because of the influence of this other place that already has an outbreak. Okay, so we can use this information in the alert system. Okay, so if one place has already a high level of cases and a huge flux to other cities, those other systems should also have a higher level of attention. Okay, this is the idea. So of course, this some of you may have already seen these slides I presented on the conference here. So of course, we have individuals interacting with each other in households, in workplaces and the education facilities, and on their leisure. So when they're going to parks, to the beaches, to shopping malls, or wherever. And this interaction of individuals in different settings is what enables the pathogen to move from one place to another. And this type of interaction does not need to take place in the same city. So an individual that lives in a given area does not need to work or study at the same area. Okay, he can be doing that in different cities. And this kind of and this type of movement is what enables a pathogen to move from one place to another. So this is why we want, and behind this is this network of mobility of individuals. Okay, and we have already some data, some census data about this, so related to how people exactly this particular interaction here. So how many individuals, an estimate actually of how many individuals live in a given city and work and or study on another one. Okay, and from that we can estimate the daily flow of individuals from one place to another, and this we can use to feed our alert system once we move to a multi-city level. Okay, so from those data we can build then this network of mobility or human mobility between cities. So let's suppose that each node here is a given city. From data, we can estimate how many individuals are moving from one place to another on a daily basis, and those flows here is what we could use in the system. So this is, and we have this data already, and now the challenge is, okay, so how we use that into the system as another level of information. Finally, last but not least, incorporating new and alternative statistical methods into our current framework. So, as Thais already mentioned, we have a big issue, which is correcting the delay in reporting cases. Okay, so what we are receiving as the number of cases in the previous week will be altered in the near future, because it's not already all the reported cases. Okay, so all the cases that occurred on that week. Okay, so we receive a given number and we have to correct that for what is the actually number of cases once all of those cases have been accounted for. Okay, so we have already a model for that, and what we want to do is enhance this model to use a dynamic one that we can change the correction parameter, let's say, that we incorporate. Because of course, as you can imagine that as the outbreak develops during the year, the difference between the number of cases that are reported and what are the actual final number of cases in the same week varies greatly along the year. It's not the same ratio for the whole year. Okay, so this is one important thing, is to have a dynamic function for that correction. Okay. Another important thing that we are currently looking into is, as Flavio mentioned, the activity thresholds and how to use historical data to define those thresholds. So when we move, for instance, from the orange alert to the red alert, for instance, okay, and how we can use historical information, which will most likely be region dependent, okay, how we can use the historical data to define activity thresholds, okay. Also do some performance analysis and improve our method based on that, okay. And another goal that we have is to, because currently what we are doing is just a nowcast, okay. So we are reporting what is the current situation. We are not saying anything of what is to be expected in the near future. And this is one of our goals as well, okay? So, of course, not a long time forecasting, because this is another beast altogether, but at least short range, okay? Next week, next 2 weeks, something like that, okay? So in the near future, what can we say about what will be the situation? So this is one thing that we are pretty confident that we should be able to tackle, and have always been a goal. Okay, so about let me just go a little bit about activity thresholds. So this is an example. This is an image taken from the article published recently by Vega and collaborators about one of the uses of the moving epidemics method, which does exactly that. So you take historical data for a given region, and from that you can, based on statistical analysis, you can define different levels of activity. Okay, you can define, for instance, what is an epidemic threshold in the sense of when typically the epidemic starts, so this rise. So when you move from just basically statistical noise to an actual exponential increase in the number of reported cases. So this is one thing, epidemic threshold. So you can use R0, RT, or look into the data and see what is the characteristic number of cases when you are in the stochastic noise region and what you have in here. You can also define what is the low level. So we have here a medium intensity level, from which between the outbreak threshold and the medium intensity threshold, this would be defined as a low intensity. This you can think of as of our orange area, more or less, okay? And above it, you can define also different thresholds, and these are all based on the historical data that you have, to define what is a high number of cases with respect to what you usually have from previous years. Okay, so this is one of the things that that we can use to better define the threshold between when should I use an orange alert or should I move to the red alert, for instance. Okay, so this is one thing. This is an example taken from this publication using influenza-like illnesses, and we have done this is one implementation of this method to our data in Rio de Janeiro city. Okay, and this is in a specific programmatic area, so it's one of those subdivisions that Thais showed us. So see, this is the programmatic area number 1, and I can never remember what number is what area, but anyway, this is not really important right now. So what you have here in different colors is the estimated incidence for each year, for each season. So in red, you have the 2010-2001 seasons, because remember, the peak is during usually in the end of summer. So we look into our data between from the winter of the previous year to the winter of the current year, because our peak then will be in the middle of this window, more or less. Okay, so this is why we have a season that is moving from one year to another, because we are looking from winter to winter. Okay, so we have 2010-2001, 2001-2011, so and so on and so forth. And so from this historical data, we come up with an estimate of what we can define as a pre-epidemic threshold. And this means that once the number of cases goes above this threshold, it means that, okay, season is started. Okay, now we are in the phase where from now on, we should expect the number of cases to increase week by week. Okay. And the same goes for a post-epidemic threshold, which means that once it crosses from up to from a value that is above this threshold to a value that is below this threshold, I can say that, okay, based on this historical data, we can expect that from now on, we will only have sporadic cases, okay, until we reach this threshold in the next season, okay? And this is what is the estimated median activity threshold. So this would mean that in this region here, I would have the orange level, let's say, and these and above, I would have the red alert. And this is tunable, okay? This is defined based, I can set what is the the percentile that I want to use of historical data to define those thresholds. So this is based on the median. This threshold here, which is the high activity threshold, so above this is not only high, but it's very high in comparison to historical data. And this is based on the 90th percentile, okay? And this is then an application to data here in HEO for a particular programmatic area. Other things that we can do, this is using, again, this same method, but taking different information. And this is now for severe respiratory syndrome. It's not SARS, mind you. The acronym is the same, but it's not the same thing. From south of Brazil. And when it's, the method is the same, but here I have then the epidemic thresholds, activity thresholds. We can see what is the typical size of the epidemic peak. Okay, this is based on the pre-epidemic and post-epidemic threshold. I can see what is the typical window of outbreak that I have in a particular area. So there are a lot of statistical tools that we can use to enhance and extract different informations from the data that we are receiving. Okay, and with this, now just let me thank not only you but also the team that is working on this project. This is, you can see, there's a lot of people, and there are also other individuals that are not named here. This is the various institutions that are taking part on this project. So, and with this, I end what I had to say here. I like to thank you all for your attention, and please, Fabio, you can take from now on. Thank you.
Yeah, now thanks Marcelo for this great talk. I think it's time for us to maybe exchange some ideas about this. If you have questions for the presenters, or, you know, let's just discuss this and figure out. This is what we came up with in the last, I would say, we're operating officially for a year, but we've been working for over 2 years on the design and implementation of these tools. We all know, I mean, all the team, we know that we are very far from an ideal situation, an ideal system, but that's, you know, that's something that I think it's a huge step from where we were before, because in Brazil, we have this, I've already said that, you know, it's very, the data is very segmented, and for us, it was only possible to come up with this alert system because we were able to get the data given to us by the city, because otherwise the data will have to go a big round trip to the central government and then come back to the public, and we will never be able to say anything timely about the epidemic. So that's I think the critical thing that made possible for us to do this. I think we have a lot of other challenges ahead of us, but we're willing to work with everybody interested to make this even better and going forward. And we also are offering, not only asking help, but we are offering access to the data that we already have. So if any of you want to look at this detailed data, you can contact us, and we are more than happy to grant you access so that, you know, we can have more heads working on this problem. So the word is now with you. If you have questions, comments.
Take a leve.
Some comment. Why not to expand this for another disease as Zika and Chikungunya, for instance?
Yeah, thank you for this question. This is one thing that I forgot to put on the presentation. And yes, we are thinking about, for instance, with what is knocking at our door is Chikungunya and Zika. I mean, this is the structure is there. I mean, it's just a matter of incorporating the data. With Twitter, we have to look how to change the parameters there, and this be with UFMG team, see how they can adapt their system of the dengue filter for Twitter to incorporate also Zika and Chikungunya. So we have to look into the symptoms, what is the difference, to see if it can differentiate from one another, and the cases that now they are collecting. So yes, this is also one of our our next steps. It is to incorporate other vector-borne diseases like Chikungunya and Zika. And of course, the same idea could be implemented to other diseases, not necessarily vector-borne. But yeah.
I would also add that it's a great luck that this disease has very weird names, because it's very easy to pinpoint in the Twitter. If they were called like table or chair, we would never be able to track them. Yeah, yeah.
Yeah, especially in São Paulo, I think.
Actually, that happened with dengue as well, because people jokes a lot. So there's a lot of ironic comments and things like that. So that's why you need a classifier that was kind of smart enough to tell apart from people joking that. But that's one of the challenges we'll have to tackle.
Oh, good to hear.
Zika can be complicated because there is well, I don't know, there is a documented case of sexual transmission of Zika. It has been occur in Colorado. Some scientists working in Senegal, and one of these guys gives Zika to his wife. So I don't know how is the transmission something, but this is a real documented case. So
yeah, that's
interesting. For the mother, can be interesting.
Certainly, yeah. We are here now with a great challenge to find ways to use mathematical modeling to determine what is to be expected in the dynamics, in the spatial temporal dynamics of these diseases, so that we can help our experimentalist colleagues to go in the field and try to verify those hypotheses and try to look for these possibilities.
Thanks. Hi. I wonder why you are kind of focused on Twitter and not, by example, in Google searches. I mean, not. I really I'm too old for all of these. But I mean, you know, I know that searches in Google have been used for flu, because you get the symptoms and look, what's going on with me.
Yeah, that's an excellent question. Every time Twitter stock goes down, we have a chill. Well, if Twitter right now, if Twitter goes bankrupt or something, we're in bad shape. But I'd like to say that, in praise of Twitter, that Twitter is one of the few, if not the only one, social network that really gives open access and real-time open access to their data. Try to search Facebook, for instance. So, and just to answer specifically to your questions, we are in contact with a colleague in the UK that is going to try and get hold of the Google Trends data. And I should point out that Google Trends data does not, as with other Google data, does not have an open API that we can query it. So that makes a bit complicated. But we thought about it, but we still have to go through other channels to reach that source of information. Yeah, real time, exactly. Twitter is real time, it's great. With Twitter, we can know right away what's going on, which is hard. Anybody else?
I wasn't here, so I didn't see all the talks. But I think one challenge that we put to ourselves was to try to make predictions and see if we are right or wrong. So that trying to answer Claudio's question yesterday, if we were able to check our own predictions and look into the future, because until now we're like looking at past data and analyzing past data. Now we are facing all these problems when we look at the future, because all this uncertainty in the data information itself. So I think the one challenge for us and for everybody that wants to participate is like to really use all our models to make predictions and compare which one wins. I think that will be the next step, I think.
Well, certainly. And to complement a comment that was made about other diseases, I'd like to say that we are very happy that the municipalities that we contact, they are very enthusiastic about our work, and by extension, all of your work, because if you look at least in Brazil, at the number of statisticians and people who can do any kinds of data analysis in the cities, at the city level, it's basically zero. They have no people to do this. And their database and their health information system is a legacy system that's been in place for over 25 years. So they're really excited that we are willing to work with them to help that. And they, if they could, and if we could, they would have us help them with all diseases. But, this is a growing movement. I think it's really great to see scientists working together with government in the control of infectious disease. So I think that's our challenge, and we should be proud that with the help of all, I should stress that, with the help of all of the output of people in our field, we've been able to prove the relevance of what we do and to be able to make a difference in the world. So I think, with this note, I think I should inform you that today we're not going to have lunch here provided by our event. Our money has just run off. So, but there's plenty of options around here. So we're all now free. Is there anyone else that wants to make an announcement or give some other information? Just anyone? No? So thank you again. Thank all of us for being here and discuss this important topics.